Hi, everyone. I'm Sara Calhoun, and I'm a data scientist in the fungal and algal genomics team at the Joint Genome Institute. In this tutorial, I will be guiding you through tools on an individual genome portal. Each genome on PhycoCosm has its own genome portal, where you can find the data associated with it and use interactive tools to analyze the genome data. So let's get started by going to the portal homepage of the model alga Chlamydomonas reinhardtii, a unicellular green alga. You can navigate to this page either by searching or browsing from the PhycoCosm homepage, or by using the portal ID, a unique alphanumeric identifier specific to each portal. I've highlighted the portal ID for Chlamydomonas in the URL address. The homepage is the primary landing page for genome portals. It provides a broad overview of the organism, including a brief description written by collaborators about the organism itself. Typically, there's an image or two that may showcase important or distinguishing morphological features. If the genome has been published, the publication will be clearly displayed on this page, and other relevant citations may also be present. A main feature of our genome portals is the navigation toolbar on the top of the browser page. This is the way to orient yourself around the various analysis tools that a portal can offer. You can see that we're on the home page, as the Home tab is highlighted. The toolbar has options to help you find things with the Search, Blast, and Browse tabs. We have tools for comparative analysis like the annotations, MCL clusters, and Synteny browser. These comparative features are covered in a separate tutorial focused on comparative genome tools. Portals have tabs for finding information through the Info tab and getting files related to the genome through the Download tab. This genome annotation was released by the JGI's PhytoZone team, and we also host this version here on PhycoCosm. The PhytoZone tab here links to the original source for the version 5.6 release of the genome annotation. We consider this an external genome because the annotation was imported rather than produced using our standard pipeline. And a short disclaimer indicating the source is provided on all external genomes. I also want to point out that there's a context-dependent Help tab. For most pages on the portal, you can click on the Help tab and it will open a unique help page associated with that portal page. There are also some portal-specific tabs, only available for certain portals, such as the organelle and K-base tabs, which I'll cover later. Now we'll go to the Info page. Compared to the home page, the Info page has more technical details about the sequencing and annotation. For projects sequenced at JGI, there will be information about how the genome assembly was produced. External projects will note the source of the data and a description if any filtering was applied. Because this annotation was not produced using our pipeline, but by the PhytoZone team, we apply filtering to get a set of gene models that is more comparable to our internal genomes. This gene set is called the Filtered Models track. Also on this page, there are tables that summarize some stats about the genome assembly and annotation. For example, we have the genome assembly size, the N50 and L50, and the number of predicted gene models. Again, we have the publication associated with the genome, and we have links to the Eco Group and Philo Group pages. Lastly, there's a short funding disclaimer. Next, we'll go over our tools for finding genes. Each genome portal has a search page where you can search for individual gene models using text keywords or identifiers. We can enter our search term in this text box. For example, I'll type in a protein name like ammonium transporter and click the search button. We can choose whether to search by keyword, or if you have a specific type of annotation, you could select that from the pull-down menu, such as Go or PFAM. We can also choose what model track to use, such as the default set, the gene catalog, or all predicted gene models for this genome. This distinction between tracks is most apparent for internal genomes. In internal genomes, many models are predicted and then filtered down into the gene catalog. For external genomes, this distinction is only relevant when filtering has been applied, but otherwise these tracks will largely be the same. When the search completes, the results are shown in a table like this, with each row corresponding to a gene model. The first column shows information about the gene, including the unique protein ID, transcript ID, location in the genome, model name, and gene track. The protein ID and transcript ID are the unique identifiers associated with a gene model. If you know the particular ID, you can also search for that using this tool by selecting Protein ID from the pull-down menu. The other columns here show the functional annotations. And if there are user annotations, they will show up in the far right column. The search results include useful links. For example, these links with the protein ID and transcript ID will take you to the protein page and transcript page respectively. Another way to find your gene of interest is to search for it using BLAST. Your query sequences can be entered here or loaded as a FASTA file. Under search criteria, you can select the alignment program and which database you want to search against. For protein sequences, you can select the full external set of proteins or our filtered set. For nucleotide sequences, there are several options, including the genome assembly, transcript sequences, and if available, you can search against EST clusters or the organelle assemblies. The green Run BLAST button will submit your BLAST search. Another useful way to find gene features is through the Genome Browser. The browser view shows where features like predicted gene models, repeats, and aligned features are positioned along each scaffold. The top of the browser viewer shows the base position along the current scaffold. Then there are the tracks, which display the data associated with this region of the genome. The first track shows the GC percentage. Next, the VISTA tracks show the conservation between the genome and another one based off of the genome alignment. So I'll scroll down the page to show you the gene model tracks, right below the scaffold track in black. Shown here in bright red, the external models track represents the gene set from the original annotation. As I mentioned, we apply filters to generate the filtered models track shown in dark red. And the gene catalog track is shown in blue. By default, tracks are shown in the condensed view, meaning that overlapping features will be collapsed down together in a single row. Many tracks can be expanded. Here we can expand the filtered models track by clicking on the plus sign next to the track label. You can also expand the track by clicking on it itself. Now that we've expanded the track, we can see individual gene models and their orientation. Below the gene model tracks, there are additional feature tracks. In yellow, there are these tracks that show BLAST-X hits against the NCBI-NR database. And in black, there are tracks showing repeat regions. Some genome portals have data beyond the standard genomic data. We include these as tracks so that we can visualize multiple data types on the browser. For this genome, we have a lot of different types of data. Sometimes it is generated at the JGI, and sometimes it's produced by our collaborators. In this case, we've imported a lot of data from external published sources. One of these tracks is the proteomics track colored here in gold. We imported a proteomics dataset from the PRIDE database and mapped it onto our filtered models track. We can see here that several gene models in this view have support from this proteomics dataset. We've also imported a dataset showing the location of single nucleotide polymorphisms from 39 resequenced laboratory strains of Chlamydia monas. This SNPs track is shown in brown. We can also visualize the locations of DNA modifications, such as N6-methyldeoxyadenosine, or M6A. Here shown in light blue, we have imported an experiment identifying the locations of M6A DNA modifications under different conditions. This can be used to validate the location of translation start sites. We can also display RNA-Seq data as tracks. Here, we've imported in purple a time course RNA-Seq experiment showing the change in gene expression under nitrogen deprivation over time. This display is an incredibly useful tool for users who have lots of different types of multiomics data and want a way to visualize this information. At the top of the browser page, there are controls for modifying the view. For example, we can pan the location shown on the browser by different lengths by using these position arrows. The current coordinates of the view are shown in this box. The format is the scaffold name and then start and end coordinates. We can type in the position that we want displayed and click Apply to directly jump to a region. The zoom controls adjust the length of the region shown on the browser. You can zoom in or out and also go directly to base level resolution or amino acid level resolution. Next, we have the general controls on the right with other options, including getting the DNA sequence of this region shown, flipping the orientation of the strand, and you can open or close the browser toolbar. The browser toolbar allows you to further customize the view. For example, you can display or hide tracks. These controls also allow us to directly navigate to a protein of interest if we know the protein ID. I'll type in the protein ID of an ammonium transporter gene that we were looking at on the search page. And I'll click Apply. When the browser view loads, it will now be centered on this gene model. And this gene model will be highlighted in yellow. We can click on the individual gene model to open up a submenu. And then clicking on this link will take us to the model's protein page. The protein page contains information about an individual gene model. The top of the page shows the name of the gene model, the unique protein ID, and its location in the genome. We also have external links to the gene model in Phytosome. And functional annotations also include some external links to NCBI and Interpro. These internal links will take us to our annotation pages. The next section has a few additional links. View Modify Manual Annotation will go to the transcript page. On the transcript page, registered users have the ability to add their own manual annotations. There's also a transmembrane domain link. It shows how many transmembrane domains were predicted using TM-HMM, and clicking this link will show more details about the annotation. Next, there are several graphics in the middle of the page. The top graphic shows the intron-exon gene structure. The coding region is shown in red, and the untranslated region is shown in blue. The lengths of the introns and exons are provided below and above the graphic. Clicking on this graphic will bring up the nucleotide sequence annotated with the coding region and, if available, the UTR sequence. I'll click back on the web browser to return to the protein page. Below this graphic, the next line shows the scale bar for the length of the protein sequence. Under the scale bar, there are annotations corresponding to the signal peptide predicted by signal P, shown here in dark purple, and protein functional annotations in gold. Then we have the protein graphic shown in green. The vertical lines show the location of the splice sites. Clicking on this graphic will bring up the protein sequence. Below the protein graphic, top blast hits are shown for this gene model, and the descriptions on the right serve as external links. And then finally, at the bottom of the page, there are tools to filter these blast hits and retrieve their sequences. So far, we've only been looking at the nuclear genome. Some genome portals have an organelle tab, which shows options for the mitochondrial genome and chloroplast genome. Here we have a visualization of the mitochondrial genome of Chlamydomonas reinhardtii, imported from NCBI. This table on the left has some interesting stats related to the annotation. If applicable, the link to the original data source is provided in the stats table. This visualization shows gene models colored by gene type corresponding to this legend on the left. Next, the black bar shows scaffolds. And further in is a line graph showing the percent GC, extending outward from 0 to 100%. GC content that falls below 25% will be shown in this blue region, and above 25% will be in green. Inside, repeat regions are shown in blue and orange connecting lines, depending on the orientation. You can click on this image to get a high-resolution version for download. Like the mitochondrial genome, we have also imported the chloroplast genome from NCBI, and the visualization is in the same format. Another newer feature of the Genome Portal is a tab that links to Kbase. For a handful of organisms, there are metabolic models that the scientific community has created. We partnered with Kbase to import published metabolic models into the Kbase platform. Clicking on this tab will take you to a Kbase narrative so you can access the Chlamydomonas metabolic model along with the corresponding genome and use Kbase tools for additional analyses like flux balance analysis. Kbase regularly hosts training events and webinars if you're interested in learning more. And this concludes our tutorial on single genome tools. We covered tools on our portal for analyzing a single genome and covered some new features that will be expanded on in the future. Thanks for listening, and we hope you'll find these resources useful for your own data exploration and research questions.